So we need that, just keep that.
Everyone, court's back in session.
Okay, I presume one of the experts will be the next witness?
Yes, Your Honor.
Okay, which one is that?
Jennifer Hornyak, Your Honor.
Okay, and, Ms. Hopkins, will you stipulate that she is an expert in her field?
Yes, Your Honor.
And I know that the state will ask about her educational background anyway, so that's fine.
And
I'll have no cross on that point. Your Honor, my expert is in the courtroom for this witness's testimony. My understanding, the state's going to take this witness, and then the other two DNA persons will probably be tomorrow.
First,
So
in the morning and tomorrow morning.
Ms. Cross will exit when their expert exits.
Okay, that's fine.
And Judge, just to let the court know, in addition to an expert in the field of DNA analysis, it would be an expert in the field of DNA evidence interpretation. It's a specialty as well.
This is
For Ms. Hornyak, DNA evidence interpretation.
that would be the primary.
That,
area of expertise.
as well as DNA, forensic DNA analysis.
DNA analysis, as well as
DNA evidence interpretation.
evidence interpretation. Okay.
All rise.
All right, please be seated. How was lunch? I thought we had gone past that. Okay, we're ready, Jim.
Your Honor, the state's next witness would be Ms. Jennifer Hornyak.
All right. And she will be testifying as an expert, and the defense has stipulated that she will be testifying as an expert, which cuts down a little bit of time in trying to qualify her. So she'll be testifying in DNA analysis as well as DNA evidence interpretation. Those are her areas of expertise.
Do you swear to tell the truth, so help you God?
I do.
Would you please state your name and spell your first and last name for the court reporter?
Jennifer Hornyak, Jennifer, last name Hornyak.
Ms. Hornyak, where are you employed?
I am employed as a DNA analyst at a company called Cybergenetics, which is a technology company located in Pittsburgh, Pennsylvania that specializes in DNA interpretation.
How long have you worked at Cybergenetics?
I have worked in my current position since 2012, so 7 years.
And what exactly are your job duties there?
My job duties are to analyze DNA data from criminal cases using the computer software called TrueAllele. And TrueAllele just separates out DNA from mixtures if more than one person touches an item, as well as performing different scientific studies to test the method, test the computer system to make sure it's working as we expect.
And have you provided us with a copy of your curriculum vitae?
I have.
And is that essentially a professional resume?
Yes.
And what type of subject areas are covered in a curriculum vitae?
It has my work experience, different certifications, different studies I've published or published studies in art and peer-reviewed journals, different other scientific studies that I've performed, the number of times I've testified in court, as well as different presentations I've given.
If I could hand this to you and ask you if this is a correct copy of your curriculum vitae.
Yes, it is.
Your Honor, at this time we would ask that Ms. Hornyak's curriculum vitae be introduced as the next exhibit and ask just to briefly publish one page.
Okay, and that's number 6.
We could have the overhead for a moment.
It's coming up.
While that's being brought up, can you explain, for those who may not have a background in the scientific field, what publications are, what it means for something to be peer reviewed?
Sure. So in science, scientists can perform a study. So they might want to research how a method works or maybe some case studies, and they'll do their experiments. They'll write up the summary of those experiments in a manuscript or, a report, and then they will submit that report to a scientific journal, which is just a collection of a bunch of different scientific papers. And part of that process is called peer review, where that manuscript is sent to 2 independent anonymous reviewers in the same scientific field, and they'll basically review the paper and its results and deem its suitability for publication. So does it make sense? Do the results, purport what, or do they say what the authors mean them to say?
And are those peer-reviewed publications relied upon and used in further research by other professionals in that field?
Yes, they are.
And I note on here that you have listed the publications that you have been a part of, is that correct?
That's correct. I currently have 2 peer-reviewed publications.
And then in addition to that, you have what are titled as research reports. Can you explain just briefly the difference there?
Yes, so the research reports are just unpublished studies that I've been a part of and written up. Most scientific papers aren't sent off for peer review. Sometimes laboratories will just do, a study to make sure that what they're doing is working as they expect. And so these are just summaries of those studies. They're just not published.
And then the next section you have listed deals with presentations. And would these be presentations to other persons in the similar scientific field?
Yes, so I've given some presentations at scientific conferences to other forensic scientists, as well as some continuing legal education or talks to lawyers about the science.
And you have, in fact, actually taught courses on this subject matter, is that correct?
Yes, I have. At Cybergenetics, we actually have a training course for other forensic scientists to learn about our software and how it works, and as well as how to use it. And I've been a part of, I think it's 13 of those courses.
And I'm going to stop there and what academic degrees do you hold?
I have my Bachelor of Science degree in biology from Duquesne University, which is located in Pittsburgh. I also have my Master of Science degree in forensic science, also from Duquesne University.
And you've stated that you've previously been qualified as an expert in courts of law on this particular subject matter, is that correct?
Yes, I have.
Does that include here in Tennessee?
Yes, it does.
Now, you mentioned that the company that you work with deals with software. Your company, does it actually do the chemical analysis with the physical DNA sample?
No, we work with the data produced by a crime laboratory from that physical sample testing. So we work with the data, same starting point where they would work to do their interpretation.
Now, have you prepared a PowerPoint presentation to assist you in your testimony with the jury to help them understand the concept of DNA and your particular expertise?
Yes, I have.
Your Honor, at this time, we would ask to introduce a hard copy of that and as the next exhibit, and we have the digital PowerPoint ready to present if the court would allow. We'd ask to move that into evidence.
Okay, that'll be number 7.
Ms. Hornyak, if you could, this is the presentation that you have prepared for presentation of this particular case. Is that correct?
Yes, yes.
Okay, and if we can move to the next slide. Can you just start off and give a simple explanation of what DNA is and explain the basics of this science to the jury?
Okay, so the human body has trillions of cells, and many of those cells contain a nucleus, which holds all of our DNA. And that DNA is stored as a chromosome, so it's kind of all wound up inside that nucleus. And if we unwind one of those chromosomes, scientists can see specific locations of DNA, or locus, noted on the right, where you can see kind of the DNA written as text in the DNA alphabet. So you can read what those what those letters are.
Ready for the next slide? If you could please explain this slide. Sure.
So in forensics, to identify people, scientists look at specific regions of DNA called short tandem repeats. And all that means is, if you think of all of our DNA as, volumes of an encyclopedia. So we have 23 pairs of chromosomes, we get half from mom, half from dad, and somewhere within that encyclopedia, or within one of those books, scientists can find a specific location of DNA. Here it's just written out as, the poem, Take Me Out to the Ball Game. The beginning of that paragraph and the end of that paragraph are the same in everybody. The only difference is there's a short word repeated some number of times in a row or in tandem, and that is what distinguishes people. So here it's the word root, it's repeated 10 times in a row, and scientists can measure the length of that paragraph to determine how many times that word is repeated. So if the word root were repeated 11 times instead of 10, this paragraph would be 4 letters longer. And that's how scientists can know how many repeats there are, and therefore what that allele value is.
What is an allele? Can you just explain what an allele is?
An allele is just that number of times that word is repeated. And we get one allele from our mom and one from our dad at every genetic location. And when you pair them together, as we'll see in the next slide, that gives us a genotype or a genetic type. And so, say for example here, at one location, you get 10 repeats of the word from mom and 12 repeats of the word from dad. So pairing those together, that 10, 12, that pair of alleles is the genetic type or the genotype at this one DNA location. And so not everybody has a 10, 12. You might have a 10, 14. I might have a 13, 15. There's about 100 different possibilities at any one location. And scientists just don't test one location, they test 15 to 20. And so, when you combine all those different possibilities, overall, a person's genotype is relatively unique.
Next slide. Could you please explain this?
So with simple DNA, like DNA from one person, so a cheek swab, a blood sample, a laboratory would take that item, that swab, they'll extract the DNA from the swab, they'll make many, many copies of it, so it can be detected by the machinery in the lab.
And when you say make a copy, is that a chemical process that copies that?
Yes, they use chemicals to make many, many, many copies of that DNA, and in a test tube, and then they'll put it through machinery that will produce a signal or evidence data seen in the middle here. And that data is reflective of the alleles present in a sample. So for one person, we get one allele from mom, one from dad. So at most you would expect to see 2 signals or 2 alleles in the data. That's kind of what we're seeing here. One signal at 10, one at 12 in the middle. There's nothing at 11, there's nothing at 14 or 15. And so of all the possibilities of that person's genotype could have been before, looking at this data, you see there's only one possibility now, at 10, 12. And so a person or a computer can then say, okay, looking at this data, what's my genetic type or my genotype at this one location? And this is just a cartoon example kind of how it works. And so the evidence genotype then is a 10, 12, and we can compare that with a reference to calculate a match statistic.
Now, when you say compare it to a reference, are you speaking if you have an object, a piece of evidence that may have DNA? You're comparing it to that reference?
Yes, so getting the DNA data from that evidence item, we can compare what that inferred genetic type is to a reference profile or a known standard.
Now, here you have an example of just one particular, the 10, 12 alleles. When you are doing a comparison between the known profile, like the cheek swab or the blood drop that's drawn from someone, are you looking just at that one spot, or are you looking at several locations?
You're looking at several locations. Now there's kits to look at 20 plus locations. It usually is 15 to 20.
Can you briefly, okay, the next slide here, okay. The DNA mixture interpretation.
So now, when you have a DNA mixture, it just means that there's DNA from more than one person present on an item. So it would be like if I touch this counter and then you would touch this counter, our DNA could mix together there if it were swapped. And so now with DNA from two people in this example, the lab process is the same to produce that evidence data. So they extract the DNA from the swab, they make all the copies, they produce that data signal in the middle. But now you can see there's three signals there, three peaks, one at 10, one at 11, and one at 12. And so now you're looking at it, okay, it's a little bit more complicated to try to figure out, okay, whose DNA is here? What are my genetic types in this data? And so let's say we know one person that contributed their DNA had that 10-12, like we saw before. Now the task is, okay, what's the genetic type of that second person? And so just looking at this data, well, we see we have a peak that's unexplained at 11, or an allele at 11. And so that second person, if we say, if there's two people here, must have an 11 allele. So writing out those different possibilities, the 10-11, the 11-11, and the 11-12, they can then be assigned some weight or probability, or how well do they explain the data, as we'll see later. And that list of those allele pairs and their probabilities, or just those weights assigned to them, is that evidence genotype. And we can compare that to a known reference and then calculate match statistics in the same way as if you had a single source profile.
We've kind of talked about the basics of DNA right now. I'd like to take you back, and if you can you explain what TrueAllele is and what is the TrueAllele system?
TrueAllele is a computer system developed by Cybergenetics, and what it does is it uses mathematics, statistics, and probabilities to separate out the contributors in DNA evidence data. So it takes those signals and applies math and statistics to it to try to figure out the genetic types of the different contributors, or separate them from one another.
Has TrueAllele's reliability been tested?
Yes, it has. There have been over 35 validation studies or tests done on the TrueAllele system to make sure it's working as we'd expect, and 7 of those have been published in peer-reviewed articles.
Can you describe what a validation study is?
A validation study is a test done of a computer system like TrueAllele or any other kind of method. It could even be done in cars to make sure the cars are working as expected, just kind of to let you know, all right, I'm putting this data in, am I getting the answer I would expect out?
Okay. And has the TrueAllele system actually been validated?
Yes, it has.
Okay. What type of agencies have used your TrueAllele method for DNA analysis?
There are 8 crime laboratories currently using the TrueAllele system in their everyday casework. There's about 4 or 5 others that are purchasing it and working to bring it online to use. It's also been used to re-examine data from the World Trade Center disaster to associate the missing people with some of the victim remains.
Have you ever used a TrueAllele computer to analyze DNA samples?
Yes, I have.
And how many times have you done that in a criminal case?
I have analyzed over 500 data in 500 criminal cases, and there's probably over 1,000 of samples, evidence samples.
And do you have any type of certification for using TrueAllele?
I do. I am a certified basic TrueAllele operator, basic advanced TrueAllele operator, and a certified reporting scientist. And those are just courses offered to teach myself and others what the results mean, how the system works, and how to use it in simple cases for the basic coursework, more advanced cases or complex cases, and then how to explain those results in court.
And obviously, as we talked about on your curriculum vitae, you have participated in scientific studies and given presentations on TrueAllele to other people in the field. Is that correct?
Yes, I have.
And you have taught courses specifically about TrueAllele to other professionals?
Yes.
Now, if you could, going further into your PowerPoint, can you explain slide number 7?
So what this slide is showing is actual data from an item in this case. It's from a window frame. And it's showing, so what we see on the horizontal axis, it says size or BPs.
And you could actually touch that screen and it will leave a mark if you need to point to something.
Okay. Well, on the horizontal axis, or the one that's for size, that's just measuring the length of that DNA paragraph. The numbered boxes on top of each of those signals or those peaks corresponds to the allele value or the number of repeats present. And then the vertical axis or RFU is just the relative amount of each of those alleles that are present. And so this is at one DNA location called VWA. And so it's just showing that there's an overall pattern, there's high peaks, there's low peaks. Every peak has a size and a height. And the computer can use all of this information, that pattern, those highs and those lows, when it's trying to solve, and figure out the different profiles or contributors that are present in this sample.
Can you explain the next slide, please?
So now what the next series of slides, the next 3 slides, is going to show us is kind of how the computer's working to figure out or separate out those contributors. So what it's doing is it's proposing an allele pair, a genotype for one person in blue, here a 16-17, those blue boxes over the peaks. And then on the next slide, it might propose maybe a second person is a 16-16. And then on the next slide, maybe a third person in purple is a 15-19. And so for each of these 3 people, it's proposing that pattern of those colored boxes. And then comparing that to the data and saying, okay, does this combination of those profiles, those genetic types, explain this data? Is it a good explanation? And well, you see those colored boxes kind of correspond to that data pattern, the tops of those peaks. And you can see this is a good explanation of that data. And so it gets a higher likelihood, and those combination of genotypes get higher probabilities. And it's not just trying this one pattern out, though. It might try out moving the orange box, say, to a 19, or moving the boxes all around. It's not just trying one good explanation. It's trying out hundreds of thousands of different possibilities to figure out the contributors to this profile or this mixture. And then the next slide shows us.
Just for a second, let me go back to that one for a second. You were talking about how the computer will go through hundreds of thousands of particular scenarios. Is that in part what the difference is between TrueAllele and a human analyst being able to interpret this data?
Yes, so the computer can try out hundreds of thousands of different patterns and compare them with the data. Whereas with the human analysis, it's harder for a person to say, okay, let me try out this one pattern. All right, try out the next pattern. And maybe an hour later, they've only tried out 10 different patterns. So the computer can do it a lot faster, and it's a lot more capable of doing that. Human analysis involves basically kind of drawing a line and saying, okay, what's above this line? I'm going to consider that in my interpretation. Everything below, I'm just not going to consider. And then trying to look at it by eye and figure it out. It's kind of if you look at a surface, with your naked eye, you say, okay, I see a tabletop. But you swab that surface, put it on a microscope slide and look at it under a microscope, you might see now, oh, there's a bunch of bacteria or something here. So the computer's like that microscope. It can look at the data a lot more in depth, whereas a person can only kind of say, okay, this is what I see on the surface.
And this may be very elementary, but what is the difference or the distinction between the higher the peak or the lower the peak? What does that indicate?
Taller peaks mean that there's just more of that specific allele present in the data, and a shorter peak would mean there's just less of that present in the data.
Okay. Now, if you could continue on.
So the computer's not just trying out, good explanations, it's also trying out ones that don't explain the data as well. And so here you can see, a lot of that tall 16 peak and that 17 peak, they're not really being accounted for by this specific proposal. And so that combination of genetic types gets a lower probability in the end. And so the computer's not just trying out all the good ones, it's trying out the good, the bad, and the ugly to figure out, who's present in this data. And go ahead.
Do you need me to go back? No. Okay. If you could explain the next section.
So trying out all those different possibilities, the computer will write down for each of the 3 contributors at each of the 22 locations tested what that genotype or that genetic type is. And so what this slide is showing us is from that data for one of those 3 contributors at that one DNA location, what that evidence genetic type is. And so on the horizontal axis, we see the allele pair values, or those are ones that it tried out that were given, more weight or a higher probability, which is the height of that blue bar on top of each of those allele pairs. And so, 1616 it tried it out, and said, okay, this explains it pretty well, like 36% probability. And there's some with a little more, some with a little less. And so for this one contributor at this one location, the computer, just looking at that evidence data, said, this is what that genotype is, those allele pair possibilities with their probability or their weights. And this is done before ever knowing any comparison reference or who it would be looking for.
So this is done on the DNA data obtained from the piece of evidence, the unknown DNA.
Correct.
You're looking at that before ever comparing it or looking at the known sample.
Correct. The computer's just looking at that data from that evidence item.
Okay. You could describe this.
Mm-hmm. And so now the computer separated out the contributors, wrote down those evidence genotypes. And now what we can do is we can calculate what's called a match statistic, or just the strength of an association between the evidence and a reference.
When you say a reference, what is a reference?
It's just a known profile. So it's some DNA from a known standard, or a cheek swab taken from a victim, an elimination, or a suspect. And so on this slide, we see those same blue bars we saw before with their different weights. And then we see it's kind of a lighter tannish color. And that just tells us the chance at random in a population that you would see a specific allele pair value. So at 1516, you have that brown bar there, and that's just the chance at random that somebody would have that specific allele pair. And then to calculate a match statistic, we can compare the height of that blue bar, or the probability of an evidence match, to the height of that brown bar, or the probability of a coincidence, and say, okay, is this person producing an inclusionary statistic, an exclusionary statistic, or we just can't tell, is it kind of inconclusive? So looking at on the second from the left, the 1616, if we would compare with a reference who had a 1616, well, the blue bar is taller than the brown bar, so that would give us an inclusionary match statistic. If we would, looking at to the left of that, the 1516, the brown bar is higher than the blue bar, so the chance of a coincidence is now higher than the chance of a match. And so that would be an exclusionary match statistic, or weight pointing against that person being a contributor to this sample.
And this conclusion, the blue bar to the tan bar, is a comparison of the, say, DNA collected from a piece of evidence to the general population. Would that be a correct way of saying it?
Yes, it's the chance so if you had a specific comparison, the chance of that evidence match, or that blue bar, that probability, compared to the probability or the chance that it was that a random person had that allele pair.
I just guess I want to make sure that I'm clear on this. It's not, the blue bar and the tan bar are not comparison between the evidence and then the known sample of a suspect.
Correct. It's the chance of an evidence match versus the chance of a coincidence is that tan bar.
Okay.
And so we can compare with anybody, not just one person. So if they're not there, they'll get exclusionary weight. If they're there, there'll be an inclusionary statistic.
Next.
And so all we're adding on this next slide is now for the first time, we're introducing the genotype at this one location of the defendant in this case. And he happens to have a 1617 allele pair. And so at this one location, we can calculate what that match statistic would be if we would compare to his genetic type. And so we take the blue bar divided by the brown bar, so the 32% divided by 14%, and at this one DNA location, that match statistic is 2. Or so it's describing the, how much more does the suspect match the evidence than a random person. So at this one DNA location, it's 2 times a match between this one location and of the evidence and the defendant is 2 times more probable than a coincidence. But we just don't test one location. There's 22 locations that are tested.
If you could explain this portion?
So all this chart is showing us is what that match statistic is at each of those 22 locations tested. So the one we were just looking at is VWA. It's at the very bottom, circled in the red box. And you see that's a little over 2. And so that same process, the computer's separating out, trying out all the different possibilities at each of these listed locations on the left, and then producing those match statistics when compared to the defendant. And since all of these locations are on different parts of the genome or on different chromosomes, we can multiply these numbers together to come up with the overall match statistic or the final number or association between the evidence and the defendant.
Next.
And so that final number is that a match between the inside window frame and Roy Coons is 175 billion times more probable than a coincidental match to an unrelated African American person. A match between the window frame and Mr. Coons is 990 million times more probable than a coincidental match to an unrelated Caucasian person. And a match between that window frame and Mr. Coons is 332 million times more probable than a coincidental match to an unrelated Hispanic person.
Why are these statistics broken down by racial categories?
So just what's traditionally done in forensics is to report the match statistic across the relevant ethnic groups for an area. So typically it's African American, Caucasian, and Hispanic, but say maybe if you were in Alaska, you might want to compare it to an Eskimo population, as that could be more prevalent there. So it's just what's traditionally done in forensic science.
Now, these numbers that you have given us, those numbers are, can you explain again what they signify?
So it's just the comparison or the association between the evidence and the reference relative to a population. So it's just the chance of that evidence match, the chance of a match versus the chance of a coincidence. It's called a likelihood ratio, or it's just that ratio of is he there versus is it somebody else.
And is there another type of calculation that is done dealing with the mathematical chance that it is someone other than Roy Coons?
Yes, as part of our process in our disclosure, we can also have the computer calculate the chance that a person who didn't contribute their DNA, so a completely random person, would give us this number or higher. So basically it's just the error rate or the chance of a false positive. What's the chance we were wrong and we produced this number, and the person didn't contribute their DNA?
And did you do that calculation in this particular case?
Yes.
And I don't believe I've introduced your report yet. If I could show this to you and ask you if this is an accurate copy of your report.
It is.
And if you could take a pen and if you could write down the statistic for the mathematical chance that it is someone other than Roy Coons.
If I could refer to my
Yes,
notes, please. Thank you.
And if I could ask that that be made an exhibit and published to the jury, Your Honor.
All right, that's number 8.
Thank you, Your Honor. Now turning for the record to page 2 of your report, where the handwritten section is, and if you could read and explain that to the jury.
So when we look at these match statistics, we'll look at the smallest number of the 3. So in this case, it happens to be the Hispanic population, or that 332 million. And so the chance that a random person would produce a match statistic of 332 million or higher is 1 in 7.11 billion. And all that means is you'd have to go through 7.11 billion people before you would find another person, just by chance, that would give you that number or higher.
And what is the approximate current population of the world?
It's about 7 billion.
Now, I'd like to ask you, Ms. Hornyak, just a little bit about the concept of DNA transfer. Are you familiar with that concept?
Yes.
First of all, before we get into that, can you talk about DNA being degraded? What causes DNA to be degraded and what effect that can have on analysis?
So what can happen sometimes when DNA is deposited on a specific region is if it's, exposed to the elements, say like sunlight or weather or chemicals, what can happen is basically instead of being an intact DNA strand, it can be broken into pieces based on these chemicals, sunlight, different effects. And so if it's left out in any specific region, it can kind of be broken up. And what happens then is sometimes in the laboratory, the chemicals they use can't really pick up those different copies of, make copies of those fragments because they're all broken up into little pieces. And so it can make analysis harder because, you're not seeing a lot of the DNA that's there.
And is there actually a difference between a reason why evidence may be stored in a brown paper bag versus a plastic bag?
Yes, so mold can also affect DNA recovery, so it can kind of overwhelm the sample. So they prefer to store things in a cool, dry place, if you will, so that way there is less of a chance of the DNA being destroyed.
So humidity, moisture, things of that nature can affect and degrade DNA?
Yes, they can.
So if something were, say, hypothetically in a steamy location, would you expect that to, over time, affect the DNA?
Yes, it could.
Now, is there any way to tell how old DNA is?
No.
Have there been any, well, let me ask you this. Is there any difference between DNA, the likelihood of DNA being transferred from one specific type of surface to another specific type of surface?
There have been scientific studies that have been done to kind of, so what DNA transfer is so maybe a person didn't directly contact a region, they didn't touch it, but maybe somebody else wiped a cloth over it and then set that cloth down and their DNA is just somewhere they didn't touch. And so there have been scientific studies done to kind of look at, okay, what's the chance if someone touches a gun and then someone wipes it with a cloth and then I set that cloth on another thing, what's the chance of that happening?
And what was basically, how were those studies conducted and what results did they discover?
One specific study by Goray, Goray, from 2010, basically studied, so he put pure DNA on a substrate. It was cotton, wool, and like a plastic to simulate the 3 types of surfaces, and then blood, and then saliva, and said, okay, if I let this dry, what's the chance if I, then touch another type of that substrate, clean plastic, cotton, or wool, what's the chance that that DNA would transfer? And he also did it just kind of touching it against it, and then applying pressure, and then friction or rubbing it against the other one. And when DNA was dry from, a soft surface like a cotton or a wool to any of the other 3, plastic, cotton, or wool, the chance was like less than 1% that DNA would transfer. Now, if the DNA was wet, it was a higher chance that it would transfer because, the moisture that was in that cloth can then more easily transfer to the other items.
And this was a study that was this a type of study that was done over a period of years, leaving things sitting out, or was it simply until the items were dry at some point?
I believe it was like within, it wasn't years, it was within a specific number of days. The article has the details of how long, but it would have been until the substrate dried, yes.
Did we not finish? You're familiar with the actual testing that's referred to as Y-STR testing, is that correct? DNA testing?
Yes.
And is it possible for an analysis of Y-STR DNA analysis to read as not having male DNA, but a Y-STR testing be able to develop male DNA?
Yes, it's possible. And what Y-STR testing is it's just testing that male chromosome. So it's just looking at that Y chromosome that every male gets from their father. So each male in a family from that paternal line would have the same Y-STR profile. And sometimes in some cases it's been shown, there was a study from 2017 where they did a bunch of case reviews. I believe the author's name was Purps, P-U-R-P-S. They studied about 267 different cases and showed, okay, there were some cases where the traditional STR testing, or kind of what we saw in the slideshow, didn't detect any male DNA. That was about 68 of those cases. But then in half of those cases, or 30-some, the Y-STR testing, or just looking at that male marker, was able to develop a profile. And this can be because the amount of female DNA or other DNA could mask that, or shadow or cover up that male contributor. Or if the DNA was degraded, it can affect those bigger markers, but that Y chromosome is relatively small, so it's less affected by that degradation.
So there would be nothing unusual for an STR analysis to have a different finding than a Y-STR analysis?
That's correct. It's possible that they could, the one could not show any male DNA and the other one would.
There are actually two separate processes. Would that be correct?
They are two different types of tests done to look at the DNA.
Did you, in addition to, did you have other items that were sent to you in addition to the sample from the window seal, from the window area?
Yes, there was, I think we had 25 items total.
And if we provide you with your report, or if you have one with you, could you share with the ladies and gentlemen of the jury what was sent to you for initial analysis?
Sure, if I could refer to my notes. Thank you. So this is in our disclosure case packet, stated June 5th, 2019, and it's on page 7. It lists the evidence items that we received. We received 2 different batches. So the first was on July 12th, 2018. We received 23 different evidence items. It includes swabs from a green karate belt. There were 3 of those. Swabs from a yellow karate belt. There are 3 different swabs of that item, so there's 6. Swabs from the inside window frame, which was the item that was in the report, we were asked to report on. Swabs from the outside window frame. Cutting of a cigarette butt. Cuttings of right breast swabs. Cuttings of ankle swabs. Cuttings of right arm swabs. Cuttings of neck swabs. Cuttings of left arm swabs. Cuttings of possible hair on blue/pink pants, swabs of the inside of the left breast bra cup, swabs of the back of a white and pink shirt, cutting of a stain on a bedspread, a separate cutting of a stain 11 on the bedspread, cutting of stain 34 on the bedspread, a swab of a stain of fabric from a box spring, and a swab of the middle of the fabric from a box spring. And then in September of 2018, we were sent 2 additional items. Was the right face swabs and the right buttock swabs. And all of that data was put into the computer, and preliminary results were produced for each of the 25 items.
And did any of your preliminary results show any connection with Roy Coons?
Other than the window frame we talked about, there was also a match between that cigarette butt and Mr. Coons.
And was your analysis able to exclude him from all of those particular samples?
From the other 23 items, yes, exclusionary match statistics were produced.
Now, you mentioned right face and right buttocks. Again, would there be anything inconsistent with your not finding Mr. Coons' DNA would that be inconsistent if a test revealed Y-STR DNA?
No, based on my review of the data from those items, there was a lot of female DNA present, so that female DNA could be masking any other contributor's DNA.
Thank you, Ms. Hornyak.
All right, Frosty.
I would request a brief recess, please.
Okay, to confer with you.
Yes.
Okay, what, 5 minutes?
10.
Okay, we're a little early, but that's okay. We'll go ahead and take a 10-minute break.
All rise.
Go, 10-minute recess.